The overall goal of this procedure is to isolate active, full-length kinesin from Drosophila embryos for single-molecule biophysical studies. The first step of the procedure is to collect embryos and dechorionate them with bleach. Next, the dechorionated embryos are homogenized and the lysates are clarified. Then, microtubules are polymerized in the clarified embryo lysate, and kinesin is stably bound to the microtubules. Finally, the kinesin microtubule complex is sedimented and the kinesin is released from the microtubules. Ultimately, the ability of the kinesin to walk along microtubules can be assessed in a single-molecule bead assay using an optical trap.
This protocol is a modification of a protocol developed by Bill Saxton for purifying Drosophila kinesin. And one of the challenges faced by many beginning biophysicists is the need for functional protein. Physicists are not usually trained in making proteins. So we developed this protocol to try and make clear and easy how it is that you can purify protein so that biophysicists will have an easy source of kinesin. Kinesin is interesting because it's a mechanoenzyme, so not only is it a protein, but it walks and it exerts force, and it's a very small nanomachine or molecular motor, and we'd like to understand how it works. And this protocol will make it possible for people to understand it better.
Generally, individuals new to this method will struggle to get enough embryos for this purification protocol. This can be achieved by carefully maintaining the flies to get 50 egg-laying cups. From around 10 grams of embryos, we'll get around 200 micrograms of kinesin at the end of this protocol.
If the flies are not laying well, you could collect 3 sets of collection plates overnight.